is Ross Breckenridge. I'm the CEO of a startup company called NANOGAP Therapeutics, and we have discovered a new class of oncology medicine. This is an entirely new type of material that's been developed by our parent company that we are using to address some of the biggest unmet needs in oncology. And our molecules have a very favorable safety profile, and we believe that we're going to be able to start dosing humans in 2021. The sort of areas that we're interested in therapeutically are some of the really big unmet needs in oncology. And KRAS tumors in particular are a huge market, and they are a third of all cancers, and they really are the third of cancers that you really don't want to get. So they include drug-resistant non-small cell carcinoma of the lung and pancreatic cancer. Pancreatic cancer is increasingly prevalent as it's obesity-related, and at the moment there is no cure, and it has a very dismal prognosis. We're also interested in brain cancer because of the mechanism of our drug that I'll talk about in a minute, but crucially, our drug will cross the blood-brain barrier, so we will be able to address a really unmet medical need in oncology, both pediatric and adult. So Ag5, our lead molecule, unlike the molecules that are currently in treatment of cancers, particularly brain cancer, is freely accessible throughout the body. And because of its mechanism of action, it really does nothing to non-cancer cells. So current treatment, for example, of cancer cells, of brain cancer, most treatments actually don't even get to the site of the cancer. They don't cross the blood-brain barrier. Ag5 freely crosses the blood-brain barrier. Once oncology drugs get to the brain, they act on normal cells as well as cancer cells, leading to problems like, for example, chemo brain, the problem caused by a drug like doxorubicin, which causes functional problems in normal neurons at the same time as killing cancer cells. Our molecule acts as a catalyst inside cancer cells. So cancer cells are affected, but in non-cancer cells, our drug is inert. And this is really the unique, crucial factor for our drug. Ag5 acts to amplify the oxidative effect of reactive oxygen species. Reactive oxygen species are present in all cells, but in cancer cells, particularly KRAS mutant cancer cells, there are very, very high levels of reactive oxygen species. Now, cancer cells protect themselves from killing themselves by reactive oxygen species with antioxidants. And lots of attempts have been made to either increase reactive oxygen species level in cancer or to decrease the effectiveness of antioxidants in cancers to kill cancer cells. These have all proved to be ineffective because of off-target effects. Inducing reactive oxygen species in non-cancer cells is bad news, particularly in the cardiovascular system, where it leads to fatal complications. Our molecule Ag5 does not induce reactive oxygen species. It amplifies reactive oxygen species effect in cells that already have high levels of ROS. So our molecule acts as a catalyst downstream of the mutant RAS. And mutant RAS is a very hot area of drug development. And there are a lot of attempts to drug KRAS. The issue is that these are highly mutagenic proteins, so affecting one mutant version of KRAS will simply select out that version and allow other mutant isoforms that the drug does not affect to take over. Our molecule acts downstream, so we're affecting all of the KRAS mutant cancers, but actually all of the cancer cells that have high reactive oxygen species that aren't KRAS mutant. And we are finding an increasingly large number of cells that are malignant, have high levels of reactive oxygen species, and drive a wide variety of cancer types. So our molecule has a very favorable safety profile. Obviously, toxicology analysis is ongoing, but what we've found is that we can give therapeutic doses of our drug for prolonged periods of time to experimental animals, And this leads to no observable adverse effects, particularly in the bone marrow. And this is really important because we foresee this drug as being given daily as a tablet form to patients when they leave hospital. And for that, we need to have a very favorable safety profile. So far, we're very happy with the safety profile that we have. In terms of efficacy, we've got good evidence from PDX models of cancer, and these are animals that have been injected with human cancer cell lines. So we're actually treating human cancer cell in the context of an experimental animal. And our molecule is highly effective in glioblastoma and also in KRAS mutant lung cancer and small cell lung cancer, where the comparator drug is cisplatin, and our drug is much more effective than cisplatin, and interestingly is more effective in metastatic cells than it is in the parent tumor deposit. And this fits with the mechanism of action of our drug, because we know that metastatic cells have a higher level of reactive oxygen species than the parent cancer. So this suggests that our drug is going to be highly effective, and we're undergoing dose-finding studies at the moment to find the most efficient way of administering our drug. But we think that we have a use outside KRAS cancer because, for example, a lot of haematological malignancies are driven by reactive oxygen species, even though they have wild-type KRAS. And this is an example of a pilot experiment that we've done in-house in our discovery labs. And we find that Ag5 is as effective as bortezomib, which is a billion-dollar drug being used in myeloma. So we think that eventually the biomarkers that we're developing for our drug for use will actually spread across all cancer types. And eventually we aim to have a biomarker-defined population really in every cancer type. So we have a very wide range of potential uses of our drug. And I haven't had time to talk about radiotherapy sensitization or of chemotherapy-resistant, chemotherapy-senescent cells. And this, for a very small company like ours, is obviously a very big bite. So our aim is to partner after our clinical proof of concept trial, which will be a first-in-human trial in patients that we're hoping to enroll in 2021. We're going to perform a basket clinical trial, so-called, using a biomarker-defined population across different histological forms of cancer, and to look at clinical and also PD markers of efficacy. And we expect to get a readout of this within 5 years. And this will really drive the main value inflection point of our company. So we have an aggressive timeline for this. Currently, work is underway to develop the manufacturing process of our drug, but also to develop biomarkers and dose finding. We are starting communication with pharmaceutical companies. We aim to have a pharmaceutical development agreement with a pharmaceutical company in a couple of years' time when we are enrolling our clinical trial cohort. We aim to file IND next year, and we're going to use the FDA as our regulatory body. So our team is small, growing, and highly experienced in drug development. So my background is as an academic physician in the UK, and I've just come back from a couple of years in the Bay Area of America, where I was a biotech CEO. Martin Page is our chief scientific advisor, who ran J&J preclinical oncology for many years. We have two very eminent scientific founders who have very well-funded laboratories in Spain, which is very helpful to us because a lot of the work that I'm talking about is going on now, even as we are raising money. Graham Martin is a board member who was CEO of Takeda Ventures for many years and brings a lifetime of R&D and deal-making experience. And Darren Bianchi represents our parent company, and he brings marketing and process development expertise. So we're raising a $3 million seed round at the moment, and as you'll see, the majority of this money will go towards data and developing and manufacturing processes. We're a lean organization spread across Europe and the US. We don't have a huge fixed cost base, and we aim to deploy capital very efficiently. So we're interested in adding to our team. We're interested in finding investors who we very much see as being partners in the development journey. And we're very excited that this new class of medicine is going to revolutionize the treatment of currently untreatable cancers. So please do feel free to contact me by email if you have any questions and if you want me to send you any more information. Many thanks.